Hello everyone, welcome back to the channel. In today's segment, we'll be looking at osmosis. We'll quickly define what osmosis is and we'll look at the effects of osmosis in living organisms. So what is osmosis? Osmosis is defined as the movement of water molecules from a region of higher water potential to a region of lower water potential through a selectively permeable membrane. So what is very cardinal for you to note is that every time you are defining what osmosis is, you have to mention the presence of a selectively permeable membrane. Now, in some rare cases, they usually ask a question which states that what are some of the factors that need to be present for osmosis to take place? Well, that is very easy. You have to mention that every time for osmosis to take place, there is need for there to be a selectively permeable membrane, and also the concentration that is involved has to have different concentration gradients. So now, as you can see here, there's a diagram. We can just say it's an experiment to show how osmosis takes place. So we have solute molecules here, we have water molecules and a selectively permeable membrane. So what is happening here is that the water molecules are going to be moving about. So this process is going to be taking place until there is an equilibrium in this whole solution. Well, now that is done, we'll quickly look at the type of solutions that we'll be looking at. So we have a hypotonic solution, isotonic solution, and hypertonic solution. Hypotonic solutions are the ones whose concentration is lower than the concentration inside a living cell. An isotonic solution is one whose concentration is equal to the concentration inside a living cell. Now, what you have to note is that isotonic solutions have got no effects on living organisms. This is because it has an equilibrium that exists already. By this, I mean the concentration in both the solutions that are involved are already equal. So no need for water molecules to travel from one area to another. And lastly, we have an hypotonic solution. An hypotonic solution is one whose concentration is higher than the concentration inside a living cell. So what are the effects of osmosis in living organisms? So we'll quickly look at the effects of osmosis in an animal cell such as a red blood cell. So when an animal cell such as a red blood cell is placed in a hypotonic solution, it gains water by osmosis. As a result, it swells up and bursts. The bursting of an animal cell due to osmotic gain of water is called cell lysis. When an animal cell is placed in a hypotonic solution, it loses water by osmosis. As a result, it shrinks and wrinkles. The shrinking and wrinkling of animal cells due to osmotic loss of water is called crenation. Osmotic loss of water by an animal tissue leads to dehydration of the animal. So we can have a demonstration of what I'm just from explaining. So this is a normal animal cell, and this is an animal cell that starts to swell, and this is an animal cell that has gone through cell lysis. So when an animal cell, to summarize, so when an animal cell gains water by osmosis, it starts to swell up. As it swells up, it's going to gain more water, and as a result, it's going to burst. And the bursting of this is known as cell lysis. And when an animal cell is placed in a hypotonic solution, it's going to lose water by osmosis. So this is another demonstration of that. Now, what are the effects of osmosis in plants? When a plant cell is placed in a hypotonic solution, it gains water by osmosis. As a result, its protoplasm swells up and it eventually starts pressing against the cell wall. The condition where the protoplasm of the plant cell presses against the cell wall due to osmotic gain of water is called turgidity. The plant cell does not burst because it has a cell wall. Remember that the other function of a cell wall it's to prevent the cell from bursting when excessive fluid of pressure builds up inside it. So this is a perfect example of that function of the cell wall. So we can have a quick demonstration of how this hypotonic solution affects the cell. So we have a quick demonstration here. We have a normal plant cell. Here, it gains water by osmosis, and then once it gains water, it starts to swell. And after it gains a lot of water, the cell wall becomes bigger and it eventually starts pressing against the cell wall. And the other thing is, when a plant cell is placed in a hypotonic solution, it loses water by osmosis. As a result, its protoplasm pulls away from the cell wall. The condition where the protoplasm of a plant cell pulls away from the cell wall due to osmotic loss of water is called plasmolysis. So this is a quick demonstration of how plasmolysis takes place. So this is a normal plant cell. When it loses water by osmosis, its protoplasm starts pulling away from the cell wall and it eventually shrinks. And lastly, when any of these animal cells are placed in an isotonic solution, nothing happens. That's because, as earlier stated, an isotonic solution is already at equilibrium state, and hence nothing is going to happen. Thank you very much for being attentive. Hope to see you again in the next